Good afternoon to all the NEET aspirants. This is your Prasanna Ma'am from NEET World, and here we are to discuss the NEET previous year questions of our major challenging and demanded chapter, that is Principles of Inheritance and Variation from your Genetics. So, we will be discussing half set of the questions today beta, half set of the questions where we will be dealing with polygenic inheritance, pedigree analysis and Mendelian dihybrid cross as such few concepts, okay, and chromosomal theory of inheritance. So, these concepts we will be highlighting and discussing half set of your previous year questionnaire, and tomorrow we will be discussing next half set of questionnaire. So, let us get started. First question is on your pedigree analysis. So, if you see here, this question is very important beta. Look at this carefully. With the help of given pedigree, find out the probability for the birth of the child having no disease, and being a carrier. In brackets he has mentioned clearly it should have a disease mutation on one allele of the gene. That means he is asking us to identify for a carrier, okay, carrier female. In which generation? F3 generation. First, looking at the pedigree only, there are key characteristic features to identify whether it is an autosomal dominant or autosomal recessive disease, or X-linked dominant or X-linked recessive disease. Have X-linked recessive disease, you can identify any one of the parent if it is affected, and in the offsprings if you do not find the disease, then it will be X-linked recessive, and affected female and unaffected male will give rise to carrier females. Look at here properly. So, looking this pedigree analysis, we can confirm, we can assume first that it is an X-linked recessive disease by having some key feature, that is in the parental generation one of the parent is affected, and in the F1 generation you can see for affected female there are carrier daughters. That means it is what, it is of what type of disease? X-linked recessive disease only will be there. What are the examples of X-linked recessive diseases? Color blindness and hemophilia, okay. So, X-linked recessive diseases, fine. So, here if you see two affected female is there, affected female crossed with what? Unaffected male in the parental generation. In the parental generation, affected female with unaffected male. So, what will be the offsprings in the F1 generation? Let us confirm. XY, and then this indicates the disease presence, okay. So, here in offsprings you can find carrier females and affected sons. Of course, in F1 generation what are the offsprings that are given in this pedigree? Only female offsprings. These female offsprings are what? Carriers, okay, carrier females. Now, if you have to see F1 generation and F2 generation. In F1 generation here it is carrier female and unaffected male is giving out what? Again carrier females. Look at this carefully. Carrier female crossed with unaffected male will give you what carrier females, and carrier females and one affected male, okay. So, here again the offsprings we have carrier females only. Now, in this F1 generation and offsprings you see same the case, carrier female with unaffected male. What will be the probability of the offsprings? Of course, female children, half of the female offsprings will be carriers and half will be unaffected. Half of the male sons, half of the sons will be affected and half will be unaffected. Okay, so this will be the probability, and of course here there is a cross between affected son and a carrier daughter. and we have to identify for the carrier females in the F3 generations. Till here our assumption is clarified that it is X-linked recessive disorder, and here also we need the final confirmation, wherein here it is a carrier female crossed with affected male. What will be the probability for what? Carrier females from the total offsprings he is asking. So look at here carefully ma. First is affected offspring, next affected son, then carrier female, unaffected son. So the same scenario wherein you will get only 1/4 of the total offsprings here. 1/4 of the total offsprings as carrier females, 1/4 of the total offsprings as affected females, 1/4 of the total offsprings as affected males or affected male sons. So here what will be the right answer ma for the question? That find out the probability for the birth of a child having no disease but being a carrier, has the disease mutation only on the one allele, mutation is only on one allele. In which generation? F3 generation. So here what answer you have to mark? Option 3, because only from the final complete offsprings it is 1/4. But then in your question, if he is clearly mentioning that find out the probability amongst the female offsprings or amongst the male offsprings, then you have to mark 1/2, because amongst the female offsprings what is the probability of being a carrier female? Half. What is the probability of being an affected female? Half as such. Okay. So understood ma? First, whenever the probability basis question comes, of course we will solve 5 to 6 set of questionnaire in our advanced revision also in our coming sessions, wherein whenever probability question is given, what you have to do is, first you have to identify whether it is an X-linked dominant, X-linked recessive or autosomal dominant or autosomal recessive disorder with some key features which I have already provided you in our conceptual class. Once after analyzing which type of pedigree it is, then you have to go through for confirmation in each and every generation. Then after confirmation it will be confirmed that it will be either X-linked recessive or dominant or autosomal recessive or dominant. And then based on the question whether he is asking you the probability to find out from the entire offsprings or only from the female offsprings or male offsprings, you have to identify. Here in the question he clearly mentioned that to identify amongst the complete offsprings. Amongst the complete offsprings, only 1/4 of the offsprings are what? Only 1/4 are carrier, 1/4 will be affected females, 1/4 will be affected male and 1/4 will be unaffected male. Okay. As such you have to identify. Okay. Next question. This question appeared in your recent NEET examination, NEET 2025. Yeah. Again second question, that is, it is very simple. Ayan, continue with the class beta. Very simple question from your dihybrid cross. Okay. So genes of R and Y, that is round and yellow seeds. Okay. Round and yellow pea seeds follow independent assortment. They follow what? Independent assortment. That is, chromosomal segregation of one allele is independent to that of the other at the time of gamete formation. So genes R and Y follow independent assortment, where homozygous round and yellow, homozygous round and yellow, homozygous round and yellow seeds are crossed with homozygous recessive wrinkled and green seeds. Homozygous recessive wrinkled and green seeds. Okay. What will be the phenotypic ratio in F2 generation? In F2 generation, what will be the phenotypic ratio? So here the parental generation is given. Always do remember the parental generation will be homozygous only. After cross fertilization of the parental generation, you will get the F1 generation as heterozygous dominant. This heterozygous dominant F1 generation is self fertilized or self pollination will be done. After that you will get 9:3:3:1 ratio, which is identified by the Punnett square, okay, where 9 will be round and yellow, 3 will be round and green, and 3 will be wrinkled and yellow, 1 will be wrinkled and green. This is the phenotypic ratio that you are going to get, okay, remember that. So here option will be option 1. Understood right now beta? Next, second question. This is direct line from your NCERT, but background knowledge is required for you, okay. Next, what is the pattern of inheritance of polygenic trait? First you have to recall what is polygenic trait. Polygenic trait is a non-Mendelian pattern of inheritance, wherein according to Mendel, one gene will be having 2 alleles which code for one character, but polygenic inheritance says that one character is coded by multiple genes. Multiple genes will control one character. Best examples for polygenic inheritance. So here what is polygenic inheritance? Multiple genes will control one character. Multiple genes will control one character. Best examples for polygenic inheritance include kernel color, okay, and skin color in humans, height or tallness in humans. All this will be the examples of what? Polygenic inheritance, where multiple genes will be coding for one character. So here what is the pattern of inheritance for polygenic? It is not autosomal dominant because it is not following Mendelian pattern of inheritance. It is not X-linked. It is not Mendelian pattern of inheritance. This shows non-Mendelian inheritance pattern. Along with polygenic inheritance, let us recall few of the non-Mendelian inheritance pattern. Okay, few of the non-Mendelian inheritance patterns include what ma? Polygenic inheritance, pleiotropy, multiple allelism, incomplete dominance, codominance. These are the patterns where it is deviating from the Mendelian's laws of inheritance. You have to remember all these 5. What are the? Incomplete dominance, codominance, pleiotropy, multiple allelism and polygenic inheritance, and examples for that, okay. Ratios also. Fine, right ma? Next. This question you look into. In a cross between male and female, both heterozygous are, both heterozygous for what? Sickle cell anemia. Both are heterozygous for sickle cell anemia gene. What percentage of progeny will be deceased? Sickle cell anemia is an autosomal recessive disorder. Yes or no? Okay. So this is autosomal recessive disorder. Autosomal recessive disorder. So here if you see the cross between male and female, both are heterozygous. Both male and female, both are heterozygous for the disease. What is the percentage of progeny which is diseased? Just cross it and see. Capital A, capital A, capital A small, capital A small, small a small. It is as simple as it is. Okay, what is the percentage of the progeny that is deceased? So amongst the complete progeny, what is the probability of the deceased offsprings? 1/4. Okay, 1/4 of them is deceased because it is autosomal recessive. It should be in what condition? Homozygous recessive condition. Okay, homozygous recessive condition. So what is the percentage? It is 1/4 or 25%. Okay. Next question you look into, the production of gametes by parents, formation of zygotes. The F1 and F2 plants can be understood from the diagram called as. So what is the clear analysis in diagram or square in which you can clearly analyze what? The cross or this F1 generation, F2 generation and the offsprings. In which diagram you will be clearly observing it? In the Punnett square, that is option A. Okay. Next, So, this questions appeared in NEET 2021. Next, identify the wrong statement with reference to the gene I that controls ABO blood grouping. Before going into the options, you should recall ABO blood grouping comes under which concept? Multiple allelism. I told you, right, multiple allelism is a deviation from Mendelian's law of inheritance. Why? Okay, ABO blood grouping comes under the concept of multiple allelism. What is multiple allelism? According to Mendel, one gene will have two alleles, but then here in multiple allelism, one gene will be having more than two alleles which are controlling for one character. Okay, here one gene and many alleles. Fine. As such, to control blood grouping for I gene, for blood grouping character I gene, this I gene will have three alleles. One is IA, second is IB, third is IO or IO. Okay. IA codes for A blood grouping, B for B blood grouping and I gene. Okay. So, these are the alleles, multiple alleles for one gene. Okay. So, when IA and IB are present together, they express same type of sugar. Wrong. Okay. This is wrong statement because IA will be having N-acetyl galactose sugar over it. Over it, it has what? N-acetyl glucosamine. N-acetyl glucosamine. Glucosamine. Okay. Whereas IB blood grouping over the surface, it has galactose sugar. IA is having N-acetyl glucosamine, IB is having galactose sugar. So, it has two different sugars. As it has two different sugars, it has two different antigens. Okay. So, IA and IB are present together, they express same type of sugar is wrong, they express two different types of sugars. B, allele I does not produce any sugar, true, allele I does not produce any sugar. The gene I has three alleles, yes, IA, IB and I, okay, where I is coding for what? O blood grouping which does not have any antigen, true, okay. This has three alleles. A person will have only two of the three alleles, yes, a person will have only two of the three alleles. As such, in that combination we have 4 phenotypes and 6 genotypes, okay, 4 phenotypes and 6 genotypes. Remember, for ABO blood grouping we have 4 phenotypes. What are the 4 phenotypes, beta? A blood group, B, AB and O, and we have 6 genotypes. What are the 6 genotypes? For A blood grouping, it can be IA, or IAIO or normal I. Next, for B blood grouping, it can be IB or IBIO. Fine. And for AB blood grouping, it will be AB blood grouping, IB. For O blood grouping, it can be II or IO. Got it? So, ABO blood grouping, which comes under the concept of multiple allelism, which is deviating from Mendelian's law of inheritance, where one gene is controlled by multiple alleles. Blood grouping, I gene is having IA, IB and IO, 3 alleles, where A blood group is coding for what antigen? N-acetyl glucosamine, B for galactose. So, it has how many? 4 phenotypes and 6 genotypes. As such, you have what? Wrong statement is asked in the question to be identified. So, option A will be the right answer over here. Next question, select the correct match. Okay. So, sickle cell anemia is autosomal recessive on the chromosome level 11, thalassemia is X-linked, hemophilia is Y-linked. This is wrong. Okay. So, here it's asked to in the question it is being asked to identify the correct match, right. So, here Sickle cell anemia is autosomal recessive on the chromosome level. Thalassemia is X-linked. Hemophilia is not Y-linked. Phenylketonuria is autosomal recessive disorder. Okay, it is autosomal recessive disorder. Phenylketonuria, sickle cell anemia, thalassemia, all these are what? Autosomal recessive disorders. So, in that case, thalassemia being X-linked will be wrong. What are the X-linked recessive disorders? X-linked recessive disorders include color blindness and hemophilia. Okay. So, examples are most important beta. What type of diseases comes under which category following the Mendelian laws of inheritance. Okay. So, here sickle cell anemia, which is autosomal recessive disorder, this is true only. Thalassemia is not X-linked, this is autosomal recessive disorder. Option B is wrong. Hemophilia is not Y-linked. Phenylketonuria is autosomal recessive. So, the correct match here is option A. Okay. Next question. Experimental verification of inheritance was done by whom? Experimental verification of inheritance is done by whom? Experimental verification of inheritance is done by Sutton and Boveri. Sorry. So, who gave us the clearance about Mendelian's law of inheritance? Chromosomal theory of inheritance is given by Sutton and Boveri. Sutton and Boveri gave us chromosomal theory of inheritance. Okay. Chromosomal theory of inheritance. Fine. And then the experimental verification for the chromosomal theory of inheritance is given by Morgan. Thomas Hunt Morgan gave us the experimental verification where the model organism is Drosophila melanogaster, that is fruit fly. Fine, right? So, here option B will be the right answer. Next question. How many true breeding plant varieties? True breeding plant varieties means what, ma? Homozygous, dominant homozygous recessive condition. True breeding plant varieties did Mendel select as pairs which were similar except in one character with contrasting traits. As contrasting traits is asked in the question, total how many characters are considered? 7 characters. 7 characters, what are they? Seed shape, seed color, pod shape, pod color, flower color, flower position and height of the plant. As such, how many? 7 characters are being considered. 1, 2, 3, 4, 5, 6, 7. 7 characters are considered. For each character, there are 2 contrasting traits. One is dominant and one is recessive. Seed shape, what is dominant beta? Round seed shape is dominant and wrinkled is recessive. For seed color, yellow seed color is dominant, green is recessive. Pod shape, what? Inflattened shape is dominant and constricted is recessive. Pod color, green pod color is dominant and yellow pod color is recessive. Flower color, purple color or violet color is dominant and white color is recessive. Flower position, okay, axial position is dominant and terminal position is recessive. Height is tall is dominant and dwarf is recessive. So, as such, how many contrasting traits are there, ma? For there are total 7 characters. For each character there are 2 contrasting traits, means there are total 14 varieties or 14 contrasting traits. So, in your question, the question is asked about characters, okay, how many characters are considered, then you have to pick the option 7. Okay, 7 characters are being considered by Mendel. But in the question he is asking you about the contrasting traits or varieties, mentioning that if the question is asked, then you have to go for what? 14 varieties in total, which is option A. Clear, right? So with this we have finished out the first set of NEET previous year question practice for principles of inheritance. Tomorrow we will complete the complete set of questionnaire practice for NEET PYQs in the chapter principles of inheritance. So that's all for today beta. Thank you. If you are having any doubts, anything with respect to this session, you can drop your message in the comment box where I will be answering your doubts. Okay, thank you.